good morning. Welcome back. I know we had a very wonderful session in the morning, but now please stay put. I'm sure you had some fruits, and therefore you're not that hungry. And this session is quite interesting, right? Now, we are having Professor Dr. Chooi-Ling. She's from Malaysia, and she is from International Medical University, IMU. Now, she's going to talk about microsporin, like amino acids, which is a natural sunscreen from the marine algae. And I think Brazilians also are surrounded by water, and maybe you'll be very interested of her talk. So, welcome, Professor Chooi-Ling. You are given 20 minutes. You're standing in the second session against lunch, so you have to keep to the time. When it's the last 5 minutes, you can either look at Prof. Viranud or Prof. Chandra and me. They will do this. High five. Then you have 5 minutes to wrap up. The reason is we would love to have the students and the postdocs and the research fellows to ask you questions. So we're allocating 10 minutes of discussion. So your presentation is 20 minutes.
Yeah, I thought speak longer so that there's no time for Q&A.
Ah, yes. All right, you can start now.
Thank you so much, Prof. Shubha. Thank you. So good afternoon, good morning, everyone. Bon dia. And I am very, very happy and very honored to be here. I'd like to give my sincerest thanks to Prof. Ana Paula Girol and, of course, Professor Viranud Misapator for the wonderful opportunity to stand here before you today. Okay, my presentation is going to be really short, unlike the previous presenter, but his presentation was amazing. Just to share with you a little bit about what the project that I'm doing right now. Okay? So we have a lot of collaborators. Some of them are from the British Antarctic Survey, and also from the UK, from Chile, yeah, and we have also collaborators from within my university. So we are a small private university. I don't have a picture of it, unfortunately, in Kuala Lumpur, Malaysia. It's a similar weather as Brazil, very hot and very rainy. Yeah. Okay. So I'm not sure whether you relate to this picture. So this is actually a type of algae. Yeah, they're sunbathing. I notice Brazilians love the sun. So when you're sunbathing, you need to have sunscreen, right? So this particular algae doesn't need sunscreen because it has natural sunscreen called astaxanthin. Okay, they turn themselves a bright red. Okay, so we can't do that. So we rely on sunscreens. So here we have this beautiful picture. All of you look like this right now. Beautiful, okay? Perfect skin. And 15 years down the road, if you don't put any sunscreen or protect your skin, what will happen? You become like this. Okay? And this is a natural aging process, but also it's accelerated by the sun. The sun's properties will actually cause your skin to age rapidly, right? So here, if you break down sunlight into the various spectrum, okay? So you have the ultraviolet spectrum. Ultraviolet itself is divided into UVA, UVB, and UVC. UVB is the one that penetrates into our skin and causes all the DNA damage that causes accelerated aging. So you have oxidative damage, basic sites, which is the DNA, the basis of the DNA getting replaced or damaged. You have mutations, photoproducts. So you have all this almost completely absorbed by the epidermis, that's the UVB spectrum. And then it forms these cyclobutane dimers, which are photoproducts. And these, in the long run, may cause skin cancers, right? If it's not just aging, it's just skin cancer. So you have all these products. How many of you use this? Not many. Okay, yeah, ladies at the back. Thank you, Prof. Deepa. Okay, so we use this. It's important. But do you know it has all these chemicals? Titanium dioxide, zinc oxide, yeah? So it's, in a way, in the long run, it's not very helpful or very healthy as well. So what do? We search in nature, just like Prof. Sunil mentioned. In nature, there are many remedies. So we have found in algae, microalgae especially, a compound called MAA. Yeah? So microalgae is nature's green gold. It can be used for consumption, yeah, for nutrients. It can also be used for various bioactive properties. So it contains carotenoids, astaxanthin, lutein, and fucoxanthin, among some of the antioxidant products that we can find from microalgae. And on the skin, because I'm particularly interested on skin, yeah, we study phototoxicity, photo damage. We actually have a UV irradiator. So we subject cells to UV radiation. Okay? So it's also found to have immunomodulatory, antimicrobial, and even anti-cancer properties. So in some of the papers, yeah, that we found, spirulina combats UV-induced skin damage, and we have this microactinium, which is an anti-wrinkle study, anti-wrinkle. So perhaps you have half the face, you put some sunscreen from this extract, half the face you leave it like this, and you look at the results later on. I'm not sure how people sign up for this study, but it's interesting nonetheless. So MAA, talking about this particular compound called MAA, or this short for mycosporin-like amino acids. It's a type of amino acid. So it is a low molecular weight, water-soluble. So when you extract MAA, you need to remember it is water-soluble, use aqueous, yeah, use water extract. And it has this absorption spectra, okay, of 310 to 365. So MAAs have been studied extensively as a natural sunscreen. So what is new then that we're introducing here? Okay, so the mechanism is just very simple. It is a protective layer on top of the skin. Yeah? And these, if you're interested in chemistry, I'm not so much into chemistry, but if you like chemistry, these are the molecular structures for MAA. There are many different types of MAA, okay? But it has this cyclic sort of structure. Now, my students did a little study on tropical microalgae. It means microalgae that you can find in tropical countries. And perhaps even in Brazil, you can find these microalgae. She subjected the cells to UV and treated the cells with microalgae extract. And this is what she found. The proliferation, that means how fast the cells divide, actually reduced extensively by UV damage. Okay? If you subject the cells to UV, it stops growing, or it grows very slowly. And then she found with the extract, it enhances, it actually rescues the cells from this UV damage. Okay? But without UV, there's no difference. Okay? So that's an interesting finding. No UV and with UV. So coming to my study itself, for my PhD student, we looked at Antarctic microalgae. How many of you have been to Antarctica? Antarctic? No? Not yet. I like that spirit. Okay, soon. So Antarctic is a very extreme climate, right? Very, very cold, extreme dryness. But in this climate, you can still find living organisms, and some of them are microalgae. So these microalgae are very resistant to cold. And when I culture these microalgae in the lab, I have to keep them in the fridge. Yeah, it won't grow well outside in room temperature. So these are unicellular organisms. They live on the sea ice. They can live in the seawater itself, or even in a permafrost, yeah, on the land. It is a valuable nutrient source for plankton. You know plankton? The small, tiny organisms that larger marine animals feed on. Okay, so this is like the bottom of the food chain. So microalgae is an important food source. And they are also bioindicators for change. What do I mean by this? Now, many researchers for environmental scientists have looked at microalgae. So they looked at how this microalgae would survive or how they react towards climate change. If there is a change in the temperature of the seawater, then the production of certain chemicals in the microalgae will change. Yeah? So microalgae is used as a bioindicator for environmental change. It also responds to UV intensity. So one of my colleagues actually studies on microalgae responds to UV radiation. So here I'm studying cells, our own mammalian cells, keratinocytes, the effect of UVB on the keratinocytes, whereas my friend studies the effect of UV on the microalgae itself. Yep, so these are some of the studies that show the impact of Antarctic ice microalgae, polysaccharides, on oxidative damage. And they found that it is useful, it reverses oxidative damage. And here you see, in the mice as well, you have this photolysed nanoliposomes that is extracted from microalgae, from Antarctic microalgae. So why do I use Antarctic microalgae especially? You may ask. We have so many abundant tropical microalgae. It's because in the Antarctic, if you study geography or environment, the intensity of the sunlight is much, much stronger than in areas like, for example, in Brazil. So the microalgae there, they have to withstand much, much stronger levels of UV. So how do they do that? And we want to find out how, okay, and apply it on humans. So here, another study, and this is my collaborator, Dr. Wong Chuyen. She was the one who started Antarctic microalgae research in my institution. And she actually studied the effects of climate change on microalgae from the Antarctic. So a little bit of preliminary study from my PhD student is studying on Antarctic microalgae. We found that, again, between no UV and UV actually suppresses. This is a cell migration or scratch assay. So how do we do it? We culture cells in a plate, right, with wells, about 6 centimeter in width, and we do a scratch in the center with a pipette tip. So after 24 hours, we see how the scratch is, and we add extracts, we monitor the growth. So the closure of the gap actually shows how fast the cells can migrate. The healthier the cells, the faster they migrate. Okay? So they found that UV actually reduces the migration rate. But unfortunately, we didn't get a very significant result with the Antarctic extracts. Okay? So we need to do a bit of optimization there. Okay, so this is, I'm coming to the end. Yeah? This is a summary of what my PhD student is supposed to do the next 3 years. So scratch assay, we have bioassays to look at the effects of the microalgae extract on skin cells, keratinocytes. So hopefully we'll be able to extract and purify certain biochemicals that is useful. Yeah, we can use that biochemical instead of having to harvest microalgae from the Antarctic, which is really difficult. So this is my PhD student on your right. Yeah, his Chia can come with us this time, and a bunch of undergraduate students who are also very, very helpful and very enthusiastic in this research. Okay, that's it from me. Thank you very much. This is my contact details. I'll be happy to answer any questions.
Thank you, Chooi-Ling.
Shortest.
Thank you, Chooi-Ling. And I welcome the audience to ask questions. Okay, Prof. Deepak.
I have a couple of simple questions. So, microsporin, right? What is the structure? And you were saying it's like amino acids. I saw one of them is glycine. Can you explain a little bit the structure of microsporin and what are the amino acids you're adding?
Okay, yeah. Thank you, Prof. Deepak. Like I mentioned, I'm no chemist, but I did show some of the. If I can go back, yeah. All of them are microsporins. All of them. They have different types of microsporins. So the R is glycine, yeah, and we have different active sites. No, these are natural compounds. No, these are natural compounds. So we don't do anything to them. No modification to that. Yeah, single amino acids. Cyclohexane ring. Cyclohexanone ring, yeah. So you can see the R is actually the... Yeah. Yeah. No, it's natural. Yes, correct. Is it this one? Okay. I think this one. Okay, that's a very good question. Yeah, I did. Yeah, I know. Yeah. Right. Okay, so I have yet to look into that, Prof. Unfortunately, I can't give you a very good answer at the moment. But yes. So yeah, I can look into this study and find out more about that. Thank you. Thanks, Prof. Yeah.
How did your colleague, first of all, got interest towards this?
Thank you so much, Dr. Chandra, for the question. So this is a bit of history, background. In the east coast of Peninsular Malaysia, there is a state called Terengganu. Okay, it's famous for its beaches. And the state of Terengganu actually had a collaboration with the British Antarctic Survey and came up with a consortium. Yeah, and the consortium is for Antarctic research. So it's for collaboration between Malaysia and Antarctica and also the British Antarctic Survey. So we have this funding, consortium funding, and very humble and very fortunate to have received the funding last year to work on Antarctic research. So she actually, Dr. Wong actually went to Antarctica under research support, a grant, fellowship under this consortium. Yeah, so if you're interested to find out more about this, I'll be happy to share. Yeah, every year they have, so I think there are about 8 or 9 universities more adding on with this signing an MOU with this consortium, the YPSM. I'll be happy to share. Yeah.
So University Malaya is actually spearheading.
Yeah, UM also is one of them.
Is one of the consortium members, and I think this is something to explore. Prof. Deepak, we can invite you, and you can actually have a discussion with the whole team and so on.
Yes.
Now,
we can look into the galactose polysaccharides.
Now
Yeah.
I would welcome the students. You can ask the question in your language, Portuguese, and we have Prof. Andrea to translate, Prof. Zelia to translate. Prof. Paula. Because I think the only reasons you're not asking is of the language, I suppose. We are now together.
That's why I'm here. Por isso que eu tô aqui. Então, se vocês tiverem
Please
perguntas em português, eu traduzo para ela e traduzo a resposta. Tá bom?
I'll try my best to answer.
Anyone? No?
Alguém?
Whatever
But
way,
that's not
so
fair because
that
you-
way we can have these interactions and understanding what we are talking. So that if you have any doubt in your mind, that will be clarified. And it's a great audience and a great support we have here.
Yes.
Perguntar em português para. Queria saber se tem também pesquisas com essas algas para reparação de dano tecidual.
Are there any other researches using this algae for tissue repairing?
Absolutely. So did you see the one of the studies on scratch assay? Yeah, skin damage and then anti-wrinkle. So one of the properties of this microalgae is the ability to induce the skin cells to repair. Yeah, so the migration of the skin cells.
Uma das propriedades é justamente essa, de reparação.
Yes. So in a way, that is actually part of wound healing. Although it's just a keratinocyte, the upper epidermal layer, But I'm sure in the mice itself, you have many layers, as I think our first presenter talked about the different epidermis, yeah, the bioskin. So yes, it's very possible, and there are researchers working on it.
Ela falou das camadas, né, que as diferentes camadas que compõem ali as epidermes, e aí que tem pesquisa sobre isso.
Thank you, thank you. So to add on that, it's not just about photoaging, phototoxicity, it's also about combating skin cancer. Because one of the properties of UV is that it damages the DNA, and in the long run, many years down the road, you may get cancer. So to protect this DNA from being damaged, I think that's also one way of preventing cancer. Yeah, so anti-cancer properties.
Good morning. Well, here in Catanduva, we have a center to treat patients with burn injuries. So do you think this algae could be used also for a new treatment for our patients who suffered from burn skin, really bad, and sometimes need tissue reconstruction?
Thank you so much for the question. Excellent question. I didn't touch on burns, but burn is a type of injury, yeah, wound on the skin. The answer is I'm not sure because it hasn't gone to the clinical phase, but in terms of wound healing, it shows that in mice, in animal models, there is some effect for closure of the wound. That could be applicable to burn injuries as well. But with burn injuries, it's a little bit more complicated because then you have, it's susceptible to infection and the inflammation that happens. So maybe there's more, using the extract plus something else to combine as a therapy. Yeah, but that could be a very good potential study. One more question. I remember you.
I will try English. You learn or you research about Brazilian tests with use the skin of a fish. The name is tilapia, to reconstruct. You know that? You have a similar
tilapia skin?
Is there any similarity comparing to your research about using the tilapia skin with algae?
Right. Thank you for your question. I remember you from the class. Thank you for joining. Yeah. From my very limited knowledge of the tilapia skin, I've watched some documentaries on this. The tilapia skin, if I'm not mistaken, please correct me if I'm wrong, is to cover the wound to allow wound healing to take place naturally. Is to prevent infection. So it's like a natural sort of dressing, natural dressing for the wound. So I'm not sure exactly whether the properties of the skin, of the fish skin itself, can induce wound healing. That I'm not exactly sure. But this is more of a protective effect. It's prevention rather than cure. Yeah, so it's slightly different, the concept, but both are for wound healing, skin healing. Yes, I've seen. Very interesting. So the burn patient actually has this tilapia skin. It's clean, it's aseptic, right? And they have this as instead of a bandage, they have it on until the new skin grows. So it's not forever. They have to remove it as a dressing. Yeah.
Thank you.
The skin, I think, it's breathable. It's just a very top layer of the tilapia skin. Yeah. Very interesting. Yes. Okay. Any more questions? Finished? Thank you.
Thank you, Chooi-Ling. Let's invite Professor Andrea to appreciate Professor Chooi-Ling.
Presentation.